Transcription begins when the RNA polymerase binds to the TATA box upstream from the transcription start point.
So the RNA polymerase will move across the DNA and start unzipping it into two strands, the template strand and a coding strand. And while it does this, it'll add complementary bases, and our T base turns into a U.
Okay, so the difference between the DNA nucleotide and RNA nucleotides is that the bases are A, U, G, C instead of A, T, G, C.
After the RNA polymerase has left our start codon, which is AUG, it'll reach the UAG stop codon at the end of our strand, and it'll stop attaching nucleotides to the template strand and stop transcription.
So after the transcription of mRNA, it might need to go through further modification. So a spliceosome might come in and remove the introns that are found in between, which are non-coding, and it'll be leaving the exons. So here's our spliceosome, and it'll be, and then we're just left with a mRNA that only has introns. Additionally, a 5' cap can be added on the 5' end of the mRNA sequence, and or a poly A tail can be added at the 3' end of the sequence.
Once the mRNA is spliced, it will leave and enter the ribosome to start translation. To start translation, the small ribosomal subunit will bind onto the mRNA upstream of the start codon. Once that happens, tRNA will bind to the start codon, and the large ribosomal subunit will join on to begin or to finish the initiation complex. tRNA lays an anticodon onto the mRNA, and then a new tRNA will enter at the A site of the large ribosomal subunit and pass its amino acid using GTP onto the other tRNA. Once the stop codon is reached, a release factor will join and dismantle the large ribosomal subunit and the small ribosomal subunit, and the amino acids have formed to make a polypeptide.